This is Biology 1, Chapter 12 on Molecular Genetics. In Part 2 of the video lecture, we will be taking a look at the structure of DNA and the folks that were involved in the discovery of the structure of DNA as well. We know that DNA is one of our nucleic acids, and from Unit 1, if you can remember back, which you'll need to, the nucleotides are the building blocks of the nucleic acids. And all nucleotides have these parts in common. They have a 5-carbon sugar, and it's a special sugar called ribose. Remember, OSE words are often sugar. It also has a nitrogenous base and a phosphorus group. There are 4 different bases in the DNA molecule. We gave some examples of these, of course, in Unit 1 as well. Adenine, thymine, cytosine, and guanine are the 4 bases in DNA. We label them A, T, C, and G, respectively. Now, it's important to note that the molecule itself, DNA, had already been discovered. The nucleic acids had already been discovered. They were discovered in the late 1800s, and the question was, now that we have the job of the nucleic acids, which is to provide genetic information, what does this molecule look like? How does it behave? How does it give that genetic information? This was already known, that the nucleotides are made up of a ribose sugar, a nitrogenous base, which are these 4 bases here, and that phosphorus group. The purines in the DNA molecule are those that have a double ring of carbon and nitrogen atoms. And structurally speaking, this gives a little bit of hint about the structure of the DNA molecule. Pyrimidines are those molecules that have a single ring of carbon and nitrogen atoms. And cytosine and thymine are our 2 pyrimidines. Now, in order for DNA to do its job, in order for DNA to decode or read the information in that DNA molecule, the cell uses the other nucleic acid called RNA. So the job of RNA is to carry out the instructions from the DNA to make proteins. So RNA is kind of like the Sherlock Holmes, the detective, if you will. RNA decodes the message that is the protein list that is to be made from the genetic information on the cell. So DNA and RNA work together. The structure of RNA, it is single-stranded. It also has that ribose sugar, 5-carbon sugar. There's phosphorus groups as well as nitrogenous bases. However, there's a difference in RNA compared to DNA. The bases of RNA are adenine, uracil, cytosine, and guanine. Uracil is a purine, meaning that it has a double ring of carbon and nitrogen atoms. And uracil is the only nitrogenous base to be found in RNA that's unique to RNA. So think uracil is unique to RNA. So if you're reading a sequence of nucleotides, you would be able to tell if you were in a DNA strand because you would see A's, T's, C's, and G's. And if you were in an RNA strand, you would read A's, U's, C's, and G's. So there are no thymines in RNA, and there are no uracils in DNA. Now, with the structure of these actual nucleotides, with the double ring and single ring molecular structure, there was kind of a hint there as to perhaps how this molecule was put together. In 1950, a biologist by the name of Erwin Chargaff, what he did was he took samples of lots of different living organisms. He even took some samples from himself and found that in any sample of DNA, whether it be from a plant, an animal, a fungus, what have you, if there was, let's say, 20% of the nucleotides were adenine, then 20% of the nucleotides in that same sample were also thymine, meaning that in the rest of the sample, there was 80% cytosine and also 80% guanine. So there was some kind of association between these pairs of letters. The amount of adenine and thymine were always equal in a DNA sample, and the amount of cytosine and guanine were always equal in the same sample. Another technique was developed thanks to the forward progressing advances of technologies at the time. After World War II, it was very important in the scientific community to start to develop different technologies, and the X-ray diffractor was one of those technologies that were developed. It was basically what X-ray diffraction is taking a picture of DNA in its crystal structure, and any crystal structure of any object. This technique was not directly used by Watson and Crick, who were ultimately given the credit for discovering the structure of DNA. It was actually used by a lady by the name of Rosalind Franklin. And what this picture showed, and this photo was called photo 51, because it was the 51st photo that she took of DNA in its crystal structure. Now, it looks a little goofy when you look in the photo. You think, well, how are you going to know the structure of something by looking at it this way? But with the pattern of dark and light banding, this picture with this X in the middle suggests that there is a helical or curved curly structure to the DNA molecule. She was trying to put together the pieces and these nucleotides and how they fit into the molecule with this X-ray diffraction technique. Rosalind Franklin was one of the premier X-ray diffractors of her time. Unfortunately, she had ended up passing away of ovarian cancer with several years of exposure to X-rays with no protection on various body parts. She ended up developing cancer from overexposure to X-rays. But she, along with Maurice Wilkins and Francis Crick and James Watson, worked kind of together but separately on this structure of DNA. These folks, again, contributed to the knowledge that we know about this molecule today. Ultimately, who received the Nobel Prize for this was Wilkins, Crick, and Watson. And the only reason why is because at the time, Rosalind Franklin had already passed away from ovarian cancer. So what they discovered was that the DNA molecule was actually like a twisted ladder, or one of those old funky windy staircases. And it was a double helix structure. And that photo 51 was part of that process in figuring it out. Francis Crick and James Watson literally had, a physical model that they were trying to put together. And Rosalind Franklin and Maurice Wilkins were kind of going on the more technology route, but they ended up coming with the same conclusion. And finally, in 1953, there was a very short write-up of the work done by Watson and Crick for their discovery of the structure of DNA. And the only reason why it was a short write-up was because there had already been published work on the proposed structure of DNA, which had been since proven to be wrong. Chemically speaking, there was a lot of mistakes. And so these guys didn't want to be accused of not knowing what they were doing. So they wrote a very short write-up, very brief. Eventually that caught on, and we now know that the sugar, the deoxyribose in deoxyribonucleic acid, and the phosphate groups make up what we call the backbone. The backbone of the DNA molecule, if you think of it like a twisted ladder, it would be like the handles of the ladder. Or if you're thinking of a staircase, it would be like the banister of the staircase. And then what you're stepping on as you're going up the DNA molecule are the bases. So the nitrogenous bases make up the interior of the structure. And what keeps these two strands together are hydrogen bonds. Remember, we talked about hydrogen bonding in the past. Hydrogen bonds are very weak, but they hold things together so that molecules can stay together easily, and they can be ripped apart easily as well. Because of Chargaff's work, it was realized that these purines and pyrimidines really did have an association together. And in the DNA molecule, adenine always partners with thymine. And the reason why that is because they share 2 hydrogen bonds between them. And cytosine partners with guanine. The reason why that is because they share 3 hydrogen bonds between them. You won't ever see an adenine and a guanine together, because in chemistry it's not physically possible for a hydrogen bond to just be hanging with its cheese out in the wind. All bonds have to be connected for a molecule to function. So A is always together with T, and C always partners with G. What was understood about this complementary base pairing? If one side of the DNA molecule read adenine, adenine, thymine, guanine, cytosine, cytosine, guanine, thymine, guanine, adenine, cytosine, guanine, thymine, thymine, okay? If that's one strand of the DNA molecule, because these nitrogenous bases have a complementary pair, or have a buddy, if you will, A always pairs with T. So if I'm standing on an adenine and I look across my DNA molecule, I will always see a thymine. So you can see here, A always pairs with T, and G always pairs with C. It makes no difference what side of the molecule these nucleotides are on. They can be on one strand or they can be on the complementary strand. It's always going to be the same pairing. This is why in a sample, the amount of adenine always equals the amount of thymine, and the amount of cytosine always is equal to the amount of guanine, because these are base pairs. So adenine and thymine are a base pair, and cytosine and guanine are a base pair. Now, in order to get all of these adenines and thymines and cytosines and guanines together, the orientation of the backbone needs to be in a certain order. In carbon chemistry, we can label and number our carbons. The top strand and the bottom strand are basically running opposite of each other. And that is so the bonding points on the nucleotides can bond at the same place for either the top strand or the bottom strand. So the top strand runs what we call 5' to 3'. The bottom strand runs from 3' to 5'. Basically, what you need to know is that each DNA strand is oppositely orientated to each other. We talked a lot about chromosomes in our last unit, but when we really unwind them and get to the actual information in there, histones are special proteins that are found in DNA. They wrap around the DNA and create what's called a nucleosome. And several nucleosomes wound together is what we call chromatin. Remember, then we can roll up that chromatin into a chromosome. And so when you unwind a chromosome into its actual strand of DNA, it's a lot of information. That's why chromosomes are wound up and packed in chromatin and chromosomes in order to move from cell to cell. This has been part 2 of chapter 12, lecture notes for DNA and molecular genetics.